Okay, today in genetics, this set of notes is going to go over Chapter 7. It's a pretty limited section, and so you will be doing more reading out of your book when you go to answer your study guide, Part 2. So one other thing that can come into play is something called multiple alleles. And so multiple alleles, so when Mendel did his work, he studied traits that only had two alleles, so like tall, short, round, wrinkled. But we know now that some traits are controlled by more than one allele, and those would be called multiple alleles. So some traits have three or more alleles. Once again, those are called multiple alleles. For instance, blood type. So blood type has three different alleles. So you have the A allele, the B allele, and the O allele. A and B are both dominant, and the O is recessive. And so later you're going to be doing some genetics problems that deal with blood types, and so I'm just going to show this to you real quick. And so it's going to tell you what your phenotypes are and then what the genotypes are that can code for that phenotype. And so then you'll show your work and then write your genotype and phenotype here. However, no matter how many alleles determine a trait, an individual can only have two alleles for a given trait. One would be inherited from each parent. Okay. Then we have something called incomplete dominance. So with incomplete dominance, there are two alleles that influence the phenotype, but neither one is dominant. So neither one is dominant over the other. So when you cross them, you get a blending of the two. So for instance, if you had red snapdragons crossed with white snapdragons, so I think I've got enough room here to work this on the paper. So we're going to say our P generation. So we're going to say red, we're going to give it the big R. And since it is not dominant over white, we're going to assign white the big R, but we're going to give it a tick mark so that we can distinguish it from the red. So our parental generation, just like we did earlier, so one parent would be a big R, big R. So he got the big R from his parent, one parent, and the big R from the other parent. And you're going to cross it with the other individual that is big R tick mark or big R tick mark. And so this parent's phenotype appeared red, this parent's phenotype appeared white. Once again, through meiosis, your gamete here would be a big R. So always make your gamete, circle it. This parent would contribute a big R, little r. Okay, so then we're going to have cross-fertilization or pollination occur. And so the generation that comes out of this, once again, is called the first filial or F1. And so this sperm fertilizes this egg, and so the individual turns out to be big R prime. Well, in this case, they did not turn out to be red because red is not dominant over white, so their phenotype here turned out to be pink. So there was a blending of the red and the white. So we're going to self-pollinate them with the same type of individual. Now, the gamete that can come out of this F1 is a big R, or he could give a big R tick mark. This one can give a big R or a big R tick mark. Okay, so we have two alleles, or two different gametes. And so to find our F2, we're going to set up a punnet. So we only need a punnet that handles two different gametes. So big R tick mark, big R tick mark. So if this sperm fertilized this egg, that would be big R. If this sperm fertilized this egg, that'd be big R tick mark. If this sperm fertilized this egg, big R tick mark, and then big R tick mark, big R tick mark. So now the genotype ratio would be 1 big R to 2 big R tick marks to 1 big R tick mark, big R tick mark. Okay, so that's your phenotype. Oh, no, excuse me, that's your genotype ratio. And then your phenotype, this is where they kind of came across something that was unexpected. So it turned out for every one red snapdragon, they would have two that turned out pink to one that turned out white. Okay, so it didn't turn, so there was no, the red did not cover up the white. All right, so that is incomplete dominance. And then our last one is called codominance. Codominance simply means that two alleles influence the phenotype and both show up in the offspring. So an example would be like a roan cow. So roan cows will have both red hairs and white hairs. So we're not going to actually do a cross on that. Okay, so that finishes this section, but I want to show you one thing first to kind of help you out on your crosses. Okay, so on this page, when you do a cross, it's going to give you up here the letters that you're going to use. So for all of these crosses throughout here, this page and this page, you go back here and refer to your genes listed here. Okay, and it tells you who's dominant and tells you which one's recessive. And then what you want to do is you want to determine, so if it's heterozygous round, homozygous tall, so this is the dihybrid cross, set up your genotype. So for instance, this would be big R, little r, and then big T, big T. And you're going to cross it with the homozygous round short. So that would be homozygous round would be big R, and then short is little t, okay? Decide how many gametes you can get out of each. That makes it so much easier. So this person can give a big R, big T, or they can give a big R, little t. This person can only give a big R, little t. So you only need to set up a punnet that handles 2 across the top and 1 down the side. So always get in the habit of coming up with your gametes and then set your punnet up to reflect that. And now you just cross, and then you can come up with your ratio a lot easier, okay? And then on the, let's see, dihybrid, and then incomplete dominance, once again, you're going to need to set you up a key up here. I would use big L and then big L tick mark, okay? And then show your work, and then write your ratio here. So you must show your work on each one. And then blood type, once again, I've given you the genotypes, and so you show your work here. And then there's a page that talks about probability, show the work. Okay? Thank you.